Hi micro folks, this is one of the final videos for our cabbage fermentation experiment. This is now day 16, so over 2 weeks after we started the experiment. So we're just looking at physically how do our different cabbage treatments appear. So remember folks, we are comparing the impact of air being present on fermentation, so that's our aerobic treatments, and then we're comparing to the absence of air, so anaerobic. And we want to remember, folks, that when fermentation first evolved on primitive Earth, Earth's atmosphere was anaerobic, right? And then because in traditional sauerkraut or fermented cabbage recipes, you usually add salt, we also wanted to compare the lack of salt, so no salt, and the presence of salt, and see if that had an impact on fermentation. So we'll try to make this short, you guys. We'll just look at the appearance and check the pH. And then after this video, I'll plate samples from each of the 4 treatments onto TSA and Sabrod's dextrose media. Remember, TSA is all-purpose. It'll select for both bacteria and fungi. The Sabrod's dextrose is going to select for fungi, which would include our yeasts and our molds, and we'll see if we have different microbial communities there. We also did gram stains, and I'll make smears from each of these. We'll gram stain them, and then we'll do one video just trying to summarize the gram stain results and the culture results. Okay, so first of all, you guys, and again, I'm learning how to use my little webcam here. First of all, folks, we'll look at the aerobic, no salt, so this was our negative control, and it's definitely looking like it's decomposing. Right, it's looking brown. It doesn't smell horrible yet, but certainly I wouldn't want to eat this stuff. It looks like there's a maybe like a little moldy scum forming on the top. And with regard to pH, you guys, it's at pH 6 right now, pH 6. Okay, after 16 days. So that was our negative control. And obviously, folks, it looks like, you know, our cabbage would have rotted. If we were relying on these cabbages, you know, to get us through the winter, we would not be in good shape. And then, folks, let's go down here. We're still going aerobic, and now we went aerobic, so the presence of air, but with salt. And it's maybe not as far along in decomposing, but it's definitely starting to look brown. Looks like it is starting to rot. Maybe not as liquefied yet. So maybe the salt, you know, have slowed down some of the spoilage microbes, but still, folks, this aerobic with salt, it looks like it's starting to rot. pH, interesting, pH is around 7, around neutral. Okay, so that was our aerobic. So it looks to me, folks, my conclusion would be that with or without salt, if you keep your chopped cabbage aerobically, you won't inhibit the spoilage microbes, and you will get spoilage decomposition. Okay, now let's go over here to our anaerobic treatments. And again, folks, remember, fermentation is a primitive way of making a little bit of ATP in the absence of oxygen. There's no electron transport chain involved. So let's look at the anaerobic treatments. And again, Earth's atmosphere was anaerobic when fermentation first evolved, you know, some 3.6, 3.8 billion years ago. So here's anaerobic with no salt. And this one to me was really interesting because I wasn't sure if salt would be required. So right now, folks, it looks to me like the anaerobic no salt, it's still preserved, right? It doesn't look like it's not brown. We aren't getting that moldy scum on top. The color's looking good, and the pH is still pretty acidic, pH 4, right? So it looks like maybe at least at 2 weeks the salt wasn't required. Okay, and then, folks, if we go down here to kind of our traditional, oops, that was anaerobic no salt, sorry folks, and then we come back down here to the traditional anaerobic with salt, and it too is looking really, really good. Just like the anaerobic no salt, the pH was 4, so we still have that really low pH. It looks like it's not decomposing. So just based on appearance, folks, and also smell, when you subjectively describe the smell of the anaerobic no salt and the anaerobic salt, they both smell like sauerkraut to me, which to me is like I feel like I'm smelling the lactic acid. So at least at 2 weeks, it looks like the salt wasn't required. However, folks, remember the salt, it creates a hyperosmotic environment, so water moves from within the plant cells to outside the plant cells, and that would help create and maintain an anaerobic environment. And also, folks, we do want to remember that salt can inhibit the growth of spoilage microbes. So maybe having it both anaerobic and with the salt, maybe this would help preserve it even longer, right? I can just hold on to these in the garage. Our wonderful garage incubator, going to be 3 digits again next week, and maybe we'll just see if the anaerobic no salt compared to the anaerobic salt, which one of those lasts longest. And then just one last one, folks. Again, this was just kind of for fun. We also did an aerobic no salt but excess water, and again hoping the water would create an anaerobic environment, and it didn't do too bad. Its pH is running around pH 3 to 4. The liquid is definitely getting cloudy, so it'll be kind of fun to see who's growing in there and compared to our anaerobic salt and our anaerobic no salt. Okay, folks, so again, what we're going to do, we're going to culture to SAB and TSA. Today we're going to be making smears and doing gram stains, and the final thing we're going to do, folks, is we're going to do serial dilutions, try to get some cell counts on these guys, and that'll be a separate video. Okay, we'll see you on the next one.